I would like to thank the organizers for giving me this opportunity to present today. I'm Chisato from Liid Pharmaceuticals. We are an early-stage biotech tackling unsynthetic nucleotide safety through a unique approach and advancing their clinical applications. Today, I'd like to introduce our brother's technology and show its effect in the CNS. The central nervous system, or CNS, has emerged as a key target for ASO therapies. A major challenge in developing CNS targeting ASOs is neurotoxicity. These toxicities are thought to arise from the two types of target effects. One is hybridization independent, caused by the non-specific interactions between ASOs and endogenous molecules. The other is hybridization dependent, which ASOs binds to unintended RNAs and trigger RNSH-mediated cleavage. Although ASOs are promising for CNS diseases, neurotoxicity prevents significant development challenges. CNS toxicity of ASOs can be broadly divided into two forms. One is acute neurotoxicity. This typically peaks about 2 hours after dosing and often resolves within 24 hours. It depends on the ASO sequence and chemistry, and possible mechanisms include disruption of intercellular calcium signaling through glutamate receptors, as well as chelation of divalent ions in CSF. The other is late onset neurotoxicity. This appears days after administration and has been associated with mislocalization of the pathway proteins P55NLB from the nucleoplasm to nucleoli, which is thought to contribute to neurotoxic effects. The other target mechanism is hybridization-dependent interaction. It has been reported that ASOs with higher Tm values tend to show increased caspase activity and toxicity. Toxic ASO also tend to reduce long-play messenger RNAs, especially with high-affinity chemistries like LNA and CET through muscular antisense-mediated cleavage. These two mechanisms are distinct, making them difficult to address simultaneously. A technology is needed that maintains on-target binding while selectively disrupting off-target interactions. How could we avoid off-target interaction and enhance target specificity? Aiton et al. Defined the binding specificity alpha S by the following equation. It is the ratio of the concentration of ASO target and target mRNA complexes to the concentration of ASOs of target complexes in this system. To achieve a higher alpha S, we need to increase the association constant Ka between the ASO and its target AT complex. And as you can see here, we illustrate this concept using an energy diagram for further clarity. Again, if you can make delta G larger, you can make ASO target complex formulation more favorable. Traditionally, this has been approached by the introduction of high-affinity units such as LNA into ASOs. However, in many cases, such modification also stabilizes off-target complexes, which can worsen toxicity. What if we could instead lower the initial energy state of ASO, introducing an energetic penalty before it binds? By doing so, we could create a thermodynamics barrier that suppresses interaction with off-target molecules while still allowing favorable formulation of the target complex. The next question is, how can we achieve this thermodynamic control through molecular design? Our solution to this challenge is the Brothers duplex or BUDO. We introduce a complementary strand to conventional single-stranded ASO, creating a partially double-stranded structure. We call the Brothers duplex or BUDO. The BUDO duplex consists of two main domains. The first is the Toho domain, which acts as a key to unlock this duplex architecture. The rest forms the duplex domains, which adds the structural rigidity and conceal the nuclear bases, thereby acting as a lock to reduce non-specific interactions with RNA and proteins. The complementary strand is what we call the brother strand, BS, and the annealing process is referred to as purification. For the BS, we utilize peptide nucleic acid PNA. Because PNA carries no charge and exhibits low protein binding properties as shown here, The resulting partially charged duplex represents a non-canonical molecule design that effectively renders the ASO stealthy. So how do we design a blue duplex? We can measure the delta G of ASO and mRNA binding from TM data. For example, with this ApoB targeting lenigapimab, delta G is minus 61.6 kJ/mol, corresponding to a very strong binding affinity. KD is 40 picomolar. This means few molecules can outcompete the target mRNA. So our goal is to adjust delta G below close to delta G 80, but still ensure delta G is below minus. To design optimal blue, we tested complementary strands of different lengths. Interestingly, between 9 and 10 mer, the delta G flip sign at 10 mer. Blue became more stable than the ASO and mRNA complex, preventing effective binding. Therefore, PNA should be shorter than 10 mer. To test this prediction, we administered the series of blue duplexes to mice, and 3 days later, we evaluated knockdown activities in the liver along with hepatotoxicity. Looking at the left panel, you can see the knockdown efficiency decreasing as the BS length increases. Interestingly, when you take a closer look at the data, we observed a marked decrease in knockdown activity between 9 and 10 mer, exactly where the sign of delta G is reversed. Also, when it comes to ALT reduction, the BS length increases, the ALT elevation gets alleviated. And the biggest change occurs between 8 and 10 mer. This is very consistent to our prediction. This slide shows how purification affects protein binding. Using mouse liver lysate, we performed the pull-down assay comparing single-stranded ASL and blue duplexes. The STS page scale shows many protein bonds with single-stranded ASL, but as the length of the BS increases, the bond intensity decreases. Therefore, purification reduces non-specific protein binding. FGMS analysis revealed that many of these proteins are essential DNA or RNA binding proteins. The sequentration of these proteins suggests a potential to reduce cytotoxicity. To test whether purification can reduce acute neurotoxicity, we administered 100 micrograms of ASO or blue duplex to wild-type mice by ICB. Acute neurotoxicity was assessed with 24 hours, and knockdown was measured after 7 days. The parent ASL caused a severe neurotoxicity with mice dying within 3 hours. In contrast, the blue duplex shows no toxicity, and all mice survived the week. Importantly, it achieved strong knockdown across the brain with over 95% knockdown near the injection site. Given the severe neurological impact of CNS, minimizing brain toxicity is essential. Purification appears to offer a promising strategy. This is our final slide. We are building a development pipeline that includes programs for neurological diseases. We believe Plaza's technology could become the standard for Antisense Therapeutics, and we welcome partners who share our vision. We will be presenting more detailed data at the poster session, so please stop by. Thank you for listening.